There are, of course, no class sections or office hours on Thursday or Friday. It's university holiday. So what that means is you're not going to get new homework this week. So the Monday, there's only one Monday discussion section, but we will hold that. And nothing new is going to be gone over. There's not a new homework. It will be held as an office hour format. So if you have questions, go prepared to ask the IAs at that. Anyone can go to it, and you can get your participation points. If you already have your max participation points and you don't have any questions that you would like addressed at an office hour, you don't need to go to it. And then next Thursday, the pre-final will be made available after class. Remember, this is just like that pre-midterm that you took, except for it's going to be an online Canvas quiz. You'll have 3 hours to do it. It's going to be, instead of 20 questions, it'll be 40 questions. And it will be open for this 48-hour period. Reserve yourself a 3-hour time slot during that period when you definitely know you're going to be feeling good, you're not going to have distractions, and you will take that exam. Again, you only need to get a 50% on it to get the maximum score you can achieve for that. Then that Sunday, after the pre-exam closes, the IAs will hold a review session. This is the day and the time in the room that we have requested. We haven't gotten official notice that that is definitely what's going to happen. We will put announcements. I'll announce in class. I will let you know for sure. But most likely that's going to be the time. Again, we will set it to podcast, and so ideally it will be available that way as well. And this, of course, is all in preparation for the final, which is during finals week on Thursday the 11th, 3 to 6, here in this room. You all should have received an email about filling out these set forms for the class as well as for the IAs. Please do that. These are very valuable to get this sort of feedback for us, and we learn a lot from what you guys tell us in these evaluations, and to really encourage the class to participate. There will be extra credit based on class participation. If 100% of the class fills these in, you all get 1 point added to your score. Could make a difference. If only 50% of you do it, you get 0.5. So encourage your friends and your colleagues to fill this out before it is due. Any questions about these? Yes? So you will have a homework 8 that will be posted after class on Tuesday. Next Tuesday will be the last class of new information. Post a homework for you to do. And then Friday will be 3 normal discussion sections as scheduled. Those of you who normally have Monday discussion sections and therefore wouldn't have them on exam week, you can go to any one of those Friday discussions where they will go over homework 8. Question?
What about Thursday? Next Thursday.
So Thursday there will be a class. It will be an in-class review. And in fact, this is what it is going to be based on. It's going to be based on what you tell me you want reviewed in class time. So starting next Monday, 8 a.m. in the morning until Wednesday, 11 a.m., email me any topics you want me to review in that class. What I do is I take all the votes, and whatever gets the most requests, I put together slides, no new material, but maybe presented in a new way or with new examples, new clicker questions, and that's what the Thursday class will then be. So think carefully about what you request. Don't say like DNA replication, right? That was a whole class. Like what specifically about DNA replication don't you understand? If it was a homework problem, a problem from the exam, or like a topic like CRISPR, let me know and I will be happy to review that if it gets the most votes. So the notes for Thursday I will put together based on what you guys send me as requests. And so make sure to use that subject line so that I know and doesn't get lost in my email. I don't know why this room hates us, but all right, question, Madeline.
So December 1st, are all of the sections held in Pacific, or like on Fridays or Mondays, Thursdays?
So the schedule is posted on the class website. It tells you exactly the time and the location. All right. Okay, so let's, thank you. You think it's loose over there? All right, well, we'll see. All right, so in class last week, our focus was on genomics, which is this genome-wide analysis of gene structure and function. And so we talked about how there have been millions of organisms that have had their whole genome sequenced, and this information is freely available at these sequence databases. I took you through several steps of examples of how you could look up your favorite gene, how you could take the sequence, find the sequence of the gene, and then also compare that sequence to anything that is available in these databases by using this kind of blast search. So you give it a query, the sequence you have, and ask, does it match or is it similar to anything else that's available in these databases? And hopefully if you did the homework, you had some practice in using BLAST to search a database for a microRNA sequence. We then talked about when you have these genomes, of course we want to go in and identify where are the protein coding genes. To do this, we are looking for the open reading frames, but also in more complicated genomes like our own, we need to take advantage of other features, such as having the copy or cDNA sequence. Remember, a cDNA sequence, you have reverse transcribed the RNA. Most of the RNA in the cell is going to be already spliced, mature RNA. And so when you sequence that and match it back to the genome, you'll see where the exons are, and then the gaps, the spaces that aren't in your cDNA, would be where the introns are. And then finally, we talked about two different methods by which we can analyze the expression of all the genes in a given sample at once, like doing thousands and thousands of northern blots at the same time. So let's review these two different methods. So the first one we talked about was microarray analysis of gene expression. And the example used here is we wanted to compare gene expression in cells that were starved, that had no serum, versus ones where we have added serum. So you can imagine different genes are going to be turned on in those two different sort of nutritional statuses. So if we were going to set up this microarray experiment, what's the first thing we're going to need for this experiment? What do we need? Lydia? Yeah, we need a microarray. And these days, we just order them from companies. And so you just have to make sure that you order the correct one, right? If you're looking at human cell culture, you want to make sure you have an array that has human genes on it. If it's mouse cell culture, you would get one that has mouse genes on it. So first thing you need is to get the microarray. And then what are you going to do? What's the next step? Yes?
Isolate the RNA.
Yeah, so you isolate the RNA from the two different conditions, and then what do you do to that RNA? Yeah, you label it during the process of what? You're copying that RNA into cDNA. So you reverse transcribe the RNA into cDNA, and then you include a different kind of fluorescence so you can keep them apart. So in this case, a green dye was used during the reverse transcription reaction of the RNA from the cells that had no serum. And in this case, a red dye was used in the reverse transcription for the cells that we did add serum to. And so then we take those differentially labeled cDNA, so we can track the sequences that came from each cell type, and then we mix them together, and then we hybridize them to the array. These arrays, remember, they have probes or DNA oligos that are covalently stuck, but each gene is at a specific position. So we know gene A is here, gene B is here. We know at every quadrant what probes for which gene is there. And then these cDNAs that we have made differentially labeled will have the opportunity to hybridize. So it's a single-stranded cDNA, single-stranded probe. They match, they will base pair, they will stick there. If we see a green fluorescence at gene A here. What is that telling you? It's telling you that there was a lot of green cDNA, which means it came from the cells without the serum. So that means that gene A is highly expressed in the cells that don't have serum, as opposed to gene B here, which is all red. So this gene was highly expressed in the cells. It made a lot of that gene B RNA, got reverse transcribed into the cDNA, including the red dye. And so we have high expression of gene B in the fibroblasts where we added the serum. And then we might see a signal that's more similar, sort of 50-50 probes base pairing. That just means that that gene is probably similarly expressed under these both conditions based on this signal here. So any questions about how you would perform a basic microarray experiment? Khalil?
The hybridized, is it like already the array has like
The probes or the oligos, yes, they are already glued onto this microarray slide. So they are permanently there, they are stuck there. But you're only going to get a signal if they base pair to either the green cDNA or the red cDNA, right? If there's no RNA and therefore no fluorescence, then it's just going to look blank. It'll be like a black spot. You wouldn't see anything because not every gene is going to be expressed, right? Madeline?
So the microarray has the DNA in it, and then we're putting the cDNA from what we made originally with the RNA, right?
Yes, exactly. So the array has those oligos, has those probes. So, yeah, see this like gray line right here? That is a probe that hasn't yet base paired to anything. And then the way I drew it here, this green is base paired to that probe, red is base paired to that probe. So they all are for gene C. They're all the same sequence of DNA probe in that spot. And then you only see that spot light up if it base pairs to cDNA from either of these two conditions.
Do we have to like reverse transcribe it because DNA can bind DNA on the RNA, right? In this case, at least.
So usually when we do microarrays, just because DNA is more stable and we also have to add the label, this is what we do. There are some forms of microarrays where that directly uses RNA and other forms. This is just one example of how microarray analysis is done. The principles are similar with other methods, but there are other methods. All right, let's try to do a clicker question. All right, so here's the gene numbers. This is the key for how the cell or how the cDNA got labeled. And so which of the following statements is supported by the above microarray results, analyzing the expression of levels of RNA from cells with and without serum? Ten more seconds. All right, final answers. All right, so a lot of us like more than one of the above. Let's go through and see if that is true. Who wants to share their thinking for any of these? Sure, Cleo.
Choose E, because like A is correct, like you can see like E7 and 11, they are all like similarly expressed.
Right, so what we're seeing here, so A says genes 3, 7, and 11 may be controlled by the same transcriptional regulators. And I think what you were saying is they all look like they're similarly expressed. How are they expressed?
Doesn't matter like with or without.
Yeah, you're getting about the same level of expression because red and green are yellow, at least in the way that this is interpreted. So they're all showing the same expression pattern. So could they be sharing the same transcriptional regulators? Yes, exactly. Not necessarily, but that is consistent with the data that you're being shown here. So yeah, so A is a correct answer. What about anyone else want to share thinking? We like more, most of you like more than one of the above. So A is a good answer. Lydia.
I think C might be correct because gene 1 is shown with serum, so it might be degraded without serum.
Yeah.
This is just
Yeah, exactly. So gene 1, what's going on with gene 1? Well, we have very high levels when serum's there, which means we have very low or no levels when there's no serum there. Otherwise, we wouldn't see this differential. So one of the reasons why we might have low levels when serum isn't there, the RNA is degraded. And so the point here is when you are looking at these expression patterns, it can be based on both transcription as well as RNA stability, right? You are looking at how much RNA is there, and that is going to be dependent on how much you make and then how quickly it is degraded as well. So C is a correct answer. What about B? Does anyone like B? Gene 2 mRNA increases upon the addition of serum. What do we think about that? Yes?
I don't think it's correct because when you have it green, it means that the expression decreases after serum addition, but the paper says that the gene of the mRNA increases, even still be lessened.
Exactly. So we look at gene 2. All right, gene 2, wow, it's really highly expressed when there is no serum there, which means that it's not expressed when there is serum there. And so this says gene 2 mRNA increases upon the addition of serum. Actually, it's the opposite. If I wrote decreases, it would have been correct. So B is not a correct answer. What about D? The mRNA of gene 10 has similar expression levels in cells with and without serum. Yes, Arya?
I didn't think this one was correct because you can't really make a conclusion based on the fact that it's similar with or without. We just know that when it has serum, it's not able to be detected. But that doesn't really necessarily mean that with it would also, or without it would be having a similar expression level.
So if we get no red or green cDNA pairing here, what does that mean? It's not expressed. So is that the same level of expression in both conditions? So not expressed or 0 is the same. Does that make sense now? Yeah. So D is correct. So all of them were correct except for B. Does everyone feel pretty good about how to interpret these types of experiments? Madeline?
So, okay, when I look at this, I want to like split it. So, like we're saying, like the left-hand side is like, but we're putting both of them, right?
Yes, yeah. Yeah, this is just the key. This is not to say this is for this experiment, this is this experiment. This means that wherever you see green cDNA, that means there was a lot of mRNA expression under that condition.
Do we have an experiment where we do something like that, or not really with the microarray?
You probably could. There's a lot of different ways you could design the microarray. We could talk about it after class if you want. All right, so then the other type of RNA expression experiment we talked about where you can sort of globally look at what's happening in a cell type or a tissue was to use RNA sequencing. So this is to take advantage of this next generation sequencing technology we talked about. And for example, suppose we wanted to look at what genes are differentially expressed in a normal colon tissue sample versus a colon tumor. So the first step here is you would isolate the RNA from both of those different tissue types. You then need to reverse transcribe that RNA into cDNA. So we're going to be doing DNA sequencing. So you need to convert that RNA into copies of DNA. And then you can add the linkers and perform the next generation sequencing that we talked about, where you will then get millions and millions of reads all happening at once. So all these fragments of DNA are being sequenced at the same time. You get a readout of what these reads or sequences are. And then you take these reads, so we call them reads, those fragments of sequence. Usually they're about 100 base pairs long, and you map them back to the genome. And so you know what genes were expressed, because the only way you would get a read is if there was originally RNA there that got reverse transcribed to make the cDNA that you use for the sequencing experiment. And so then you map these back to the genome, and it's very powerful because it gives you information not only about the level, so how much RNA was there that got converted to DNA for our sequencing, but also which specific sequences are represented in the RNA. Remember we said we could see things like alternative splicing, alternative polyadenylation, just looking at where these reads map in the genome. And so where we left in class last week was this iClicker question. So let's go ahead and do this again, and then we'll go through the answers. So which of the following conclusions is consistent with the below RNA-Seq results? And so this is actually how those reads look when you map them to the genome. They're usually not little discrete boxes, but you're getting thousands and thousands and thousands of sequences that pile up primarily on the exons. Did you have a question?
Yes. The RNA sequencing detects the level of expression, meanwhile the array just detects the expression, whether it's expressed or not.
Depending on what probes you have on your microarray. So you could design probes that could detect different exon-exon junctions and actually see alternative splicing with a microarray. That's how we used to do it. More commonly, we now use RNA sequencing. All right, 10 more seconds. Final answers? All right, again, more than one of the above. Let's go through these. Which one of more than one of the above does somebody like? Madeline.
So I like A because I was looking at the V volume on the right.
Always pay attention to the scale. Sometimes the way that the computer will spit this out on your browser is it will bring all of them to the highest peak, but that doesn't mean it's the same number. And so here, what looks to be the same size is actually half as much as what you're seeing down here by paying attention to the scale here. So Madeline said is gene X mRNA levels are higher in old worms. And you gathered that because, well, they look similar, but this one's twice as much. And so the expression level, here's where we're getting information about expression, is higher in the old worm. So A is a correct answer. Any others? Do we like any others? Khalil.
B is also correct, because you can see like another isoform, like towards like the end, the right side, between these two molecules.
Yeah, so B says 2 isoforms of gene X are expressed in old worms. Okay, so we're looking at old worms. And so we see, so here's, of course, the exons that are annotated. We see relatively similar heights of peaks for all these exons, except for what's going on here. We see a lot less at this position. We see some, so what that probably means is a small percent of the time, we do have a spliced isoform that includes all the predicted exons, but also some high percentage of the time in the old worms, this exon is being skipped because the read level is much lower, meaning there was not a mature messenger RNA that had that exon in it. And so you're getting a lot of reads where it was actually skipped. And so we see this specifically in the old worms. Young worms, that peak is pretty similar to the other exons around it. So it is a difference between the young and the old worms. So B is correct. What about C? C says young worms alternatively splice gene X. Well, after going through B, what do we think about? Do we see evidence for that happening in the young worms? No. Pretty much every exon that is called is similarly represented in the young worms. If it said old worms, yeah, we are getting alternative splicing here. That's how we are getting these two different isoforms. So C is not correct. What about D? D says a new isoform of gene X mRNA is present in old worms. So when we're talking about a new isoform, what that means is a form of the gene that had not previously been predicted. So when you map this to the genome, remember we already have an annotated genome. So these blue boxes here, these exons, these were already there. This isoform is known. And in fact, this is the only isoform that came up in the genome before we did our experiments. So are we seeing a new isoform in the old worms? Yeah, we just said that we saw evidence for this exon being skipped a high percent of the time in the old worms. And so what that probably means is that there is an isoform that has this exon, this exon, this exon, this exon, not that one, and then this exon. And so this would be a new annotation we would then need to put on the genome, and maybe hadn't been discovered before because people hadn't sequenced from really old worms to identify this. Madeline?
So is that just kind of like noise on like in between the last two peaks there? Because I was thinking like that might be any type of, like right there.
Yeah, so there's probably less efficient splicing out of this intron. It's slower in the old worms, and so there's going to be some unspliced RNA that ends up getting copied into cDNA.
And then do you take into account like the peak there in the same one that we were looking at for B? Would that be like half of, because it's like 500, so it's like in reality it looks smaller, but it could be like 500 because the scale is different?
Well, so the scale here would be like all of the highest peak for all of these would be about 500. And so this jaggedness, that's very typical. So there's a little bit of bias to how well fragments get sequenced. So we wouldn't think anything alternatives going on there. Yes, and there's computational models that like spit out actually like what these are or not. But so this was actually an experiment that people in my lab did, and so this was really surprising to see that there might be this new isoform. And so there was actually a way that we could independently validate that there are RNAs that have this sequence, basically having the sequence of missing that exon. And that was taking advantage of this new sequencing method I told you about, this Nanopore direct RNA sequencing. So for this, you don't fragment the RNA. In fact, you don't even turn into cDNA. You just feed the RNA through these special pores, and there's a current based on each nucleotide that goes through the pore. That current is read as a nucleotide. It can identify which nucleotide is producing that current. And so you get a direct sequence of the RNA as it goes through there. And because we don't need to fragment it, when you get the entire sequence, you can read it and see, was that exon there or not? And so by doing this, we were able to validate that there are a lot of this messenger RNA produced in old worms that are actually missing that exon. So it was indeed a new isoform. All right, and then the last thing we didn't get to in class last week was Another example of how transcriptomics, basically the ability to sequence the entire sample of RNA, actually was used to identify the SARS-CoV-2 virus. So as we all remember, back in 2019, there were patients showing up with this respiratory illness. It didn't seem like, and they weren't testing for any known virus or bacteria that we knew about. And so what researchers did is they isolated fluid from these patients, imagining that it probably had the pathogen in there. Many lung viruses are RNA-based, so they decided to sequence all the RNA that was present there. And so you extract the RNA, reverse transcribe it into cDNA, so we can do our next generation sequencing. And then, of course, because this is coming from a patient, you're not only going to have pathogen, but you're going to have human DNA in there as well. So you can use computational methods to just filter out. It's kind of like a blast, find everything that matches human, get rid of that, and then take the remaining sequences and piece them together. And is there anything that we can discover with this new sequence we see? Basically asking, does this sequence now look like any previously sequenced viral genomes? And so if you want to ask that question, what would you use? BLAST, exactly. So they did a BLAST search. Would you do a protein BLAST or nucleotide BLAST? Nucleotide, right? You have this RNA sequence, you do a nucleotide BLAST, and that's exactly what they did, and that's what allowed them to identify that it was a new virus, but it was a type of virus we know about. It belonged to the coronavirus family. And that's because once they had the sequence, they could, it came back, there was all these alignments to known coronaviruses. And what was pretty amazing was that, there was a human coronavirus that had been identified before back in 2003. That's the original SARS-CoV. But the way that see how this is very distant from the SARS-CoV-2? What that means is that this new SARS-CoV-2 sequence is much more similar to coronaviruses that have been isolated from animals than this original one that had been found in humans decades before. And so I think another really amazing thing about this example is, December 2019 is when the illnesses first started to be realized that it was something new. The sequence of this new virus, the identification, was already identified and published just a few weeks later. And this just really shows the power of how quickly this sequencing can be done. Having these databases available allows you to go in and quickly identify what we are dealing with. And so this is then a good lead into what the general topic for tonight is, which are viruses, which certainly are parasites, and we think very bad things about viruses most of the time. But they also have been quite useful for teaching us many things about molecular biology, and more recently being used as vehicles to perhaps introduce genes to be used in gene therapy. And so I'm just going to give you a broad overview of the general viral life cycle. All viruses need to be in a host cell in order to replicate and to do damage. A virus that's not in a cell is harmless. And so we'll look at what these viruses need to do, why they need the host cells. We'll look at the wide variety of different genomes viruses have, and then talk a little bit about some host cell interactions. So all viruses need to get inside a cell one way or another. The example I'm showing you here are Bacteriophage. Remember, phage are just viruses that specifically infect bacteria, and they infect very specific types of bacteria for each phage. And in fact, these particular phage, they themselves, the whole virion, doesn't need to go into the bacteria cell. All it needs to do is send in the instructions for making more phage. It will inject its DNA into the bacteria cell. And then the virus will use the host cell machinery to make more viral proteins. So viruses don't have their own ribosomes. they need to use the host cell's translational machinery to make the viral proteins that they'll need. They're also going to need to make more copies of their own genome, right? So their whole goal in getting a cell is to propagate and make more of themselves. So they're going to need to make more of their viral genome. In some cases, they encode the proteins needed to copy and make more of their own genome. Some viruses use almost all of the host factors in order to copy and make their own genome. Once there's enough genome and new viral proteins that are available, they will assemble into new virions, and then they need to escape from the cell. And sometimes they lyse, sometimes they bud out, sometimes they stay in the cell for a while. But that is how they propagate, infect new cells, make more copies of themselves. So while we're talking about phage, by studying these phage and understanding that they specifically infect bacteria cells, we can start to make useful things from phage. And so here's an example, sort of a random array of different foods I have up here. Anyone know what is in common with all of these things? Not salmonella, Listeria. So they have all been parts of Listeria outbreaks. So Listeria is caused by a bacteria, little guy shown here. It's a very serious infection. Something like 20 to 30% of people who have a Listeria infection die. Right now, there's a recall going on with packaged pasta. I think 7 people have died already. I'm sure you've probably heard about this. Most of them have been recalled. This is why we have an FDA, to make sure when we find out there's these hazardous things, we get those off the shelf in an efficient way. Last year, there was boar's head meat from one particular factory. I think 10 people ended up dying from that. The worst recent case in the US, there was a farm in Colorado that had infected cantaloupes. 33 people died from that. These farmers were actually held criminally negligent because of the sanitation level of their facility. Raw milk, something like 5% of all raw milk that's ever tested always tests positive for Listeria. Most raw milk is not tested, so take your chances. And so why am I talking about this bacteria and like all these foods that would be really terrible if you ate them if it had a Listeria infection? Does anyone know what this is? Any guesses? What do you think can kill that bacteria really easily? A phage. So a specific phage that infects Listeria and not your good microbes in your microbiome has been commercially made and actually is used to spray foods. You can spray this phage on your food and eat it. Do you have to worry about it hurting yourself? No. Bacteriophage do not attach to your own cells. They can't do anything to animal cells. They only can infect bacteria and only the specific bacteria. So Listex is a phage that only can do even certain strains of Listeria. And so, question?
Sorry, so do they like get in by like a substrate? Do they mimic it, or how do they attach to the bacteria?
Yeah, so there are certain protein-protein interactions in the phage and what's on the cell wall of the bacteria, and that's how it knows this is my target. Another really important use of phage that is really UCSD is actually becoming a leader in this is the idea of using phage to treat other types of bacterial infections that harm us. So phage therapy you may have heard of. There's a lot of hope for this as an alternative to antibiotics. So antibiotics are usually broad range, kill many different kinds of bacteria, microorganisms. That's why you don't feel so well after taking antibiotics. Your microbiome has sort of been decimated. But phage, remember, are super specific. So if we can isolate and identify phage to especially go after some of these antibiotic-resistant bacteria we have now, they're not resistant to phage yet, then this may be a way of dealing with this epidemic that we're having. All right, so now let's talk a little bit about viral genomes. As I mentioned, they come in many different flavors. So some viruses have a double-stranded DNA genome. just like our own. We have a double-stranded DNA genome. At some point during their life cycle, remember, they're going to have to make more copies of that genome, but they're also going to have an RNA transcribed from that DNA, because it's the RNA that will be translated into protein. And by convention, we call the RNA that gets translated from a virus the plus strand RNA. So the plus strand would be the one that starts as the ATG and encodes whatever viral proteins are made. There are also viruses that come in as single-stranded DNA. Some are plus strand, some are minus strand. At some point during their life cycle, they will become temporarily double-stranded, and that is so that they can be transcribed to make an RNA, that is the plus strand RNA to make the viral protein. We also have RNA viruses. So some RNA viruses are double-stranded, so at some point during the life cycle, the plus strand will be used for making translation machinery. The other strand will be, they both will end up being templates to make more of the viral genome itself. There's other viruses like SARS-CoV-2 itself, poliovirus. These are plus strand, single-stranded RNA viruses. So as soon as these RNA viruses come in the cell, already have the messenger RNA, they can go right to the translation machinery, start pumping out viral protein. They also will need to make a minus strand. Why do they need to make the minus strand? It will be the template to make more plus strand, right? So this would be copied to make plus strand, which would then be packaged to make more viral genomes. We also have some viruses like flu virus, influenza, Ebola virus. They are minus strand RNA viruses. So when they come into the cell, they first need to be transcribed into the plus strand before that they can be translated and start making viral proteins. And finally, we have some viruses like HIV that are single-stranded RNA when they're in the viral genome. But when they come into the cell, they have a reverse transcriptase activity, copies the RNA into DNA, eventually double-stranded DNA. And in the case of HIV, this double-stranded DNA actually integrates into the host cell genome. And then once it's in the genome, it can be copied into RNA, and that's how you end up with more of the genome for HIV virus. This can be transcribed to make the plus strand RNA. All right, so the point of this slide is not for you to get confused and memorize all these different viruses and the types of genomes that they have. I just want you to appreciate that viruses have almost any kind of nucleic acid genome that you can imagine. And instead, what I want you to understand about this is the two things that's in common. What do all viruses need in order to make more viruses? Arya? They will need the plus strand RNA synthesized at some point in their life cycle to make what? To make viral proteins. Plus strand RNA to make viral proteins. And what else does every virus need? Yes? So it will need the host cell machinery, like the ribosomes, to make the proteins. But to make a new virus, we're making viral proteins. What else do you need? You need the genome. You need a viral genome. You need some way to make more copies of that viral genome, whatever it is. If it's double-stranded DNA, double-stranded RNA, it needs a mechanism that sometimes is largely dependent on host cell factors. Other times, the virus does it all itself to make. Way. We can talk more after class if you'd like. But some of that is sort of a philosophical more question of like, why are viruses? Why do we have viruses, right? We definitely know why they persist, right? Because they're very good at making copies of themselves. If they're not, we don't see them anymore. They don't exist, right? Madeline?
When the phage or the virus takes over, is the bacteria making its own DNA at the same time and competing to use the machinery?
Sometimes, but most of the time the viruses are very good at taking over the cellular machinery for their own uses and eventually just killing the cell. We'll look at an example of that in a few slides. All right, so let's talk first about this virus that impacted so much of our lives over the past 5 years or so. And so this is this virus, SARS-CoV-2, which stands for Severe Acute Respiratory Syndrome Coronavirus 2. Infection with this virus causes COVID. This is the disease, coronavirus disease. And so this is one of these single-stranded RNA viruses. It still very much is circulating. It's caused over 7 million deaths in the world. And this is increasing still every day. So the more we learn about the virus, the better prepared we are for blocking it, for helping patients who do develop infections and not leading to more severe outcomes. So I'm going to take you through some of the key steps for how SARS-CoV-2 infects a cell, but it's basically the same idea of any virus. It needs to get into a cell, needs to make more copies of its genome, needs to make viral proteins, package up into new variants, and get out of the cell. So this SARS-CoV-2 virus, it has proteins outside on its outside shell. Spike protein, you may have heard about, is one of these proteins that help it attach to specific cells in our body that have a receptor. That receptor isn't normally there just for the virus to come in. That receptor responds to cellular proteins and is important. The receptor can have a different sequence or abundance in different people. This is one of the reasons we think why some people are more susceptible to infection than others. It is abundant in our respiratory system, lungs, but more recently we found also that these receptors are in heart, kidney, intestines, and so that's why sometimes the virus can go all over a body. Once the virus gets into the cell, remember it's a plus-strand RNA genome, so it immediately hops onto a ribosome, starts translating, starts making some viral proteins. Some of those viral proteins are then used to make more copies of the viral genome. It's plus-strand RNA genome, so it needs to make a minus strand to be the template to make more plus-strand. Make a lot of plus-strand to make more RNA that can be translated, and then also that will be packaged as the genome. So once there's sufficient protein and viral genomes, the virus will assemble and then be released from the cell. So this kind of comes back to a question that Madeline had. What happens in the host when these viruses get inside of the cell? So normally, we have immune systems that recognize when there's foreign and something bad going on, and so you usually have an immune response to try and block this infection. However, SARS-CoV-2 is really tricky. One of the first proteins that it expresses when it gets into the cell actually does a lot to block host gene expression, which would include turning on the immune response. So it makes a protein that causes global translation reduction. It inhibits translation of host mRNAs, causes degradation of host messenger RNAs, and then it even also blocks the nuclear pores to inhibit the export of host mRNAs. All right, so that's a good way to block the host from expressing genes. But some of you should be thinking a little bit, well, hold on a second. All right, I know the virus replicates in the cytoplasm, so it's fine to block nuclear export. But if we're causing global translation reduction and degradation of mRNAs, how does the virus avoid that block, right? And the answer is we don't know yet. So this is something that is being intensively studied as We think the virus RNA actually has sequences or structures in it that help it overcome this translation inhibition that its protein does, that the host cells are subject to. Somehow it avoids degradation. We don't actually know how it's managing to do that. And so the virus has figured out a way to really outcompete the cell to have those ribosomes and the translation machinery all for itself to make a whole bunch of proteins that can then help it make more of its own genomes. All right, let's do a clicker question. So based on your knowledge of the SARS-CoV-2 life cycle, which of the following steps would make good drug targets to prevent COVID-19? Ten more seconds. All right, let's see what your answers are. More than one of the above again? How many times am I going to put this as the answer, huh? Let's see. All right, who wants to share their answers? Yes, Claire. Yeah, so we'll go. So C, neutralize virus with antibodies. This is the whole reason we get vaccinated, to make those antibodies so they recognize this foreign virus that's in you, attack it, send it to the immune system. It will never get in your cells. You won't get an infection. So neutralizing the virus with an antibody is a really great strategy. B says block binding of spike protein to cellular receptors. So remember when we went through and said that the virus needs to get in the cell, spike protein is what it uses to associate with the cellular receptors. If you block that interaction, virus isn't going to attach to a cell. It's going to be harmless. Question?
If you were to mess with the cellular receptors, would that also mess with the hormones that are associated with those receptors?
So if by blocking it, you actually put the inhibitor on the cellular receptor, that could be potentially problematic so that the receptor would not be able to do its natural response. If you put a free protein into the cell that mimics the interaction it would make with that receptor so that it would bind up all the spike protein on the outside of the virus, then you're targeting the virus, and so presumably wouldn't. We would test and make sure it doesn't have any effect. But so B and C are good answers. Do we like anything else or not like anything up here? Khalil?
I mean, A and D, we do that. We're basically like the regular transcription.
All right, so A says inhibition of RNA-dependent RNA polymerase activity. What does that mean? What is an RNA-dependent RNA polymerase? So it makes RNA, it's an RNA polymerase, and what is its template? RNA. Do we have RNA-dependent RNA polymerases as part of our? No, we don't. The virus has an RNA genome, so it needs to make an RNA-dependent RNA polymerase to copy its own genome. Using inhibitors to RNA-dependent RNA polymerases actually is one of the drugs that we do use in COVID infection because we don't have that type of polymerase in our own cellular machinery. So A is actually a good option and one that is used. D says inhibition of host translation machinery.
I thought you wouldn't want this one because, like, similar to what we were talking about earlier, it could potentially harm the regular, like, translational systems, like humans. So you wouldn't want to, like, inhibit it.
Yeah. We don't want to generally inhibit the host translation machinery. We need the host translation machinery to keep our cells alive and make all the cellular proteins. So there's actually a lot of different therapeutics that have been and are being developed based on what we know about the virus. And the key is always to think about what the virus needs that's different from the host cell. Question?
If it was a minus strand, would the RNA
Yeah, so minus strand also need to have an RNA-dependent RNA polymerase because they need to they need to transcribe that RNA template to make the positive strand. Yes. Any RNA virus is going to need an RNA-dependent RNA polymerase to make more of its genome. Question?
So literally the virus just kind of like shows up and attacks with nothing but its own genome, and then it just hijacks everything?
It hijacks what it needs to make more of itself.
But it doesn't have like any machinery whatsoever?
Oh, no, so it doesn't have the whole translation machinery, but some of them have almost everything they need. Like SARS-CoV-2 actually has pretty much everything it needs except for the translation machinery. It stays in the cytoplasm. It does all its copying in there. There's some viruses that go into the nucleus and integrate in the host genome to use the host cell machinery in order to replicate and make more copies of itself. So it's very virus dependent. But yeah, so you probably heard about the Merck pill. So this actually is a nucleotide analog that inhibits that RNA-dependent RNA polymerase. The Pfizer pill, Paxlovid, this actually inhibits a protease that the virus needs in order to mature proteins that it needs for its replication cycle. And then, there's a variety of other things that have been used. These two are very popular because they're pill form. Cheap, easy to take. Convalescent plasma, so taking plasma from someone who has recovered from COVID, that's very expensive, very difficult, although can be very effective in some cases of patients. And so, of course, we've learned a lot about this disease and the virus, but there's still a lot we really don't understand. One of them being, why do some people get infected but almost have no symptoms while others of us can be sick for weeks and have very terrible symptoms and, in fact, sometimes even die? You hear about long COVID, so we've only known about this disease for 5 years now, so what does that mean? What are going to be the long-term consequences of being infected or multiply infected with this particular virus? When we get vaccinated or have the actual infection ourselves, we build up some immunity. How long does that last? What kind of variants are going to continue to come out in the population? Will the immunity be effective against that? And then, of course, what have we learned from this pandemic? And so it's very likely this isn't going to be the first time that we face such a new virus. And so hopefully we've learned some things and we will deal with it better. And I think one of the amazing things that did come out of this is that it proved to us that we can make a vaccine very quickly using this RNA technology just by knowing the sequence of the virus. And so we learned a lot just through that. And of course, this has helped us get on the other side of this virus and this infection on many levels. And we know vaccines can be very effective because we have the example of a vaccine that's been around for a long time and basically almost put an end to polio. So polio, of course, is this devastating infection. Something like 20 to 30% of people who get infected with polio can end up with some level of paralysis. The problem is you usually starts off in your intestinal cells, but then can spread through your body, get into your nerve cells, and that's why you end up with paralysis. And fortunately, Jonas Salk came up with a vaccine in the 50s and 60s, and you can see the levels of infection and death just plummeted once this started to become widely available to people. And he actually didn't patent it because he said this was so important for all of humanity. Who could patent this? It'd be like patenting the sun. So this also shows us the great progress. So polio used to be endemic to almost all countries. And now, 35 years later, we've come close to being wiped out, although there's becoming sort of a resurgence in the past few years. In fact, we even saw our first case in the United States in decades happen a few years ago. You may have heard that wastewater in New York State tested positive for polio, and they actually tracked it down and found a person who had polio infection. He ended up becoming paralyzed. So once you have the infection, there's actually no treatment for it. So you want to avoid ever having the infection. And of course, vaccine is the way that most of us avoid that. There's a reduction or there's an uptick in what used to be mostly preventable diseases, and there's really a lot of different features that are contributing to these problems. And so you future scientists and doctors, this is something you will have to think about and deal with, and you should be aware of this. So why is polio so terrible for our cells and our nerve cells? What does it do to the host cell? It's a very evil virus. So it actually encodes a protein that as soon as it comes in the cell, it attacks and cuts in half this cellular factor, EIF4F. What is that? What is that protein? What process is it involved in? It's an IF. It's an initiation factor, right? So the little e stands for eukaryotic initiation factor. 4F is just the type it is. So it cuts this initiation factor in half. Wait a second, I thought we said that the virus needs the host cell translation machinery. What is it doing? Destroying a key factor that is needed for translation. Turns out that the virus has a trick. So poliovirus is a plus, here's its plus strand, here's the 5' end. In its 5' untranslated region, 5' UTR, there's a specific sequence and structure the RNA folds back on itself that we call an iris. This stands for internal ribosome entry site. This sequence and structure in the viral mRNA is able to recruit all of the translation factors it needs without needing this initiation factor. So it's able to bring in the 40S with its initiator tRNA, some of the other initiation factors that it hasn't chopped in half, and it binds directly into this region to position the ribosome to start scanning. This is why we call it internal. Instead of starting at the 5' end, the external part, and scanning through, it is an internal ribosome entry site. So the virus doesn't need EIF4F. Chops it in half. What does that mean for the host cell translation? Host cell messenger RNAs aren't going to be translated. Almost all of them do need this initiation factor. Virus doesn't have to compete with the host cell messenger RNAs. Does another thing, very dramatic, cleaves TBP. What is TBP? TADA binding protein. What do we need that for? Transcription by one polymerase, 2, 3? All three of them. So Pol1, which makes your ribosomal RNAs. Pol2, which makes messenger RNAs. All right, so it's good to get rid of that. But Pol3 makes tRNAs. Again, isn't the virus going to need tRNAs and ribosomal RNAs? It is, but turns out those types of RNAs are very stable. So once the virus gets in the cell, there's already a ton of tRNAs, ton of ribosomal RNAs. By the time it has gotten done making all of its protein it needs, it doesn't need more ribosomal RNAs, more tRNAs to be made by the host. basically blocks off all host protein transcription. So the virus really takes over that cell, uses this translation machinery to make as much protein, a new viral genome, as possible. And this is how it kills your cells, including your nerve cells, that will then lead to paralysis. All right, but there's a few good things about viruses. So by studying them, we have learned a lot, and we've learned how to engineer them for useful medical purposes. So we actually talked about, remember SV40 was this viral double-stranded DNA genome when we talked about which molecular process? What is going on here? This is DNA replication, exactly. So remember, this is a double-stranded DNA virus, goes into the host cell nucleus, and it uses all of the host cell replication machinery except for its own helicase. It brings its own helicase in, that large T antigen. Remember I said don't remember that name, that's the name of the viral protein. But it brings in its own helicase, and so much of what we have learned about the process of DNA replication came from studying this double-stranded DNA virus. Here's another example. So scientists were studying this adenovirus, and they knew the sequence of the genome. And what they did is they had isolated RNA from cells infected with this virus. And then they sort of used that RNA, they kind of labeled it and used it as a probe to detect where in the genomic DNA this RNA was coming from. And when they did that, so this is the real data, but here what you see is, well, there's parts where the red RNA probe is base pairing, but then there are these huge looped out sequences of the DNA. What is that? What will not pair to the mature messenger RNA? Introns, exactly. This is one of the first ways that introns were discovered. Scientists say, whoa, there's this RNA from the virus that does not match to all the regions in the genomic DNA. What are these sequences that are getting removed? And so that's one of the ways we first discovered splicing. We also can use viruses as gene therapy vectors, and there's actually examples of these used in the clinic now. And the idea here is that you have a virus and you strip out all of the dangerous protein-coding genes and then insert the human gene that you want to produce in the patient. For example, remember when we talked about this SMN1 gene that is deficient in patients that have spinal muscular atrophy? These are these poor children who don't survive long beyond birth because all their muscles atrophy because they can't produce enough SMN1. And so scientists have put that gene into a viral vector and programmed that particular virus to mostly be able to target and infect nerve cells where this protein is needed. And this is having some great efficacy in patients who are now getting viral delivered gene that they were missing. Question? Yeah. So a lot of it depends on how they're packaged. So some viruses, in a very broad sense, many viruses have just a protein shell. Other viruses have a lipid shell. And so that will definitely change a lot about how stable that virus can be outside of the body. Usually the lipid ones survive really well, and those are going to be mostly your airborne ones. Protein shell ones usually are pretty unstable and need to always be infecting cells. And then also, there'll be other proteins that stick out of even the lipid or that and will determine the tropism or which types of cells or even which types of organisms that the virus can actually infect. And then finally, and this will be a lead-in to the class we'll have on Tuesday, there are a lot of connections between viruses and cancer. So there are viruses that can cause cancer or contribute to cancer development. And there's two general ways they do that. Some viruses actually encode a cancer-causing gene. We call these oncogenes. Basically, the virus wants to program the cell to start dividing so that it can use its replication machinery to copy its own genome. And so they will somehow overcome the cell's ability to regulate cell division. And so the virus itself can bring in an oncogene. The other option is sometimes the virus will activate a host cell, what we call proto-oncogenes. So proto-oncogenes are normal genes in your genome. They are genes that promote cell division, but in a very regulated manner. When you lose that regulation, maybe because a virus has inserted in the promoter and taken out a repressive element, and so now this gene is going to be highly overexpressed, that human gene would convert from a proto-oncogene to what we then call an oncogene. It has become unregulated. And viruses, this is an advantage to the virus because usually it turns on the cellular replication machinery that helps the virus replicate its own genomes. And here's some examples I'm sure you have heard of before, viruses that are associated with human cancers. Cervical cancer, estimates are over 99% of all cervical cancers are associated with infection with human papillomavirus. There's a vaccine. You can avoid infection and avoid many of these cervical cancers. Liver cancers, infection with hepatitis B virus can turn the infected cells into tumor cells. There's also a vaccine for hepatitis B virus. Some of these others, though, we do not have vaccines for, and we know that they are associated with these various types of cancers. So we will talk more about oncogenes and tumor suppressors and cancer in the class when we come back next Tuesday. Have a great break. Think about questions that you might want me to go over in the final class on Thursday. But don't email them to me until that window of next Monday through Wednesday, because I would hate to lose them in my swamp of email.